The overall goal of the MicroSecure embryo vitrification method is to provide a safe, secure, simple yet effective aseptic closed vitrification procedure for human embryos.
This method was developed to minimize the quality control variables in the vitrification of human embryos, thus ensuring reliable and reproducible success.
The main advantage of this technique is that it strives to eliminate technical variations between biologists and laboratories.
Our technique is so simple to perform that we find the biggest challenge for our trainees is learning how to pipette embryos without losing them.
The idea behind our method was to eliminate the need for specialized devices by adapting two commonly used FDA-approved items, an internally labeled ionomeric storage straw and a sterile flexipet.
Visual demonstration of this technique is extremely helpful, as it highlights the basic tips that ensure its successful application.
Before beginning the procedure, place the sterile 0.3 milliliter semen straw onto the Teflon surface of the electrode of a basic tabletop impulse sealer just in front of the plug to create an internal seal. Depress the handle to activate the heat, releasing the handle when the red light is off and a beep is heard. Then push the cotton plug forward against the inner seal before completing the setup. For cryodilution and blastocyst loading of the samples, remove the patient culture dish from the incubator and confirm all specimen identifications with a secondary source. Under a stereo microscope,